Floating Disk Photosynthesis Lab by Lab-Dannia Kamakh. The purpose of this lab is to observe the impact that light has on the rate of photosynthesis in spinach leaves by measuring the rate of production of oxygen in uniform disks. When the disks are placed under dark and light environments without oxygen, the disks will sink in solution. The disks in the cup that are exposed to light will produce oxygen faster and start to float again faster than the disks that aren't exposed to the lights. First, we must gather all the materials needed for this lab. You will need 8 grams of baking soda, or NaHCO3, dish soap, 2 large syringes, 3 beakers. One of them should be 500 milliliters, and then the other 2 should be 200 milliliters. A Sharpie marker and tape for labeling the beakers, 2 large spinach leaves, tweezers, a hole punch, distilled water, lamp, aluminum foil, stopwatch, graduated cylinder, a weigh boat, scapula, stirring rod, pipette, and an electronic scale. Prepare the soap and water solution by measuring 20 milliliters of distilled water and adding soap until the solution is green in color. This solution will be used later on in the experiment. Prepare a sodium bicarbonate solution by measuring 8 grams of baking soda on a weigh boat that is zeroed out on the electronic scale. You will need to measure 150 milliliters of distilled water in a graduated cylinder. Next, in a 500 milliliter beaker, pour the distilled water and pour the sodium bicarbonate while mixing the solution. Ensure that the sodium bicarbonate is completely dissolved and that there are no clumps at the bottom of the beaker. Using a single hole puncher, make 20 leaf discs. Make sure you avoid any veins in the leaves. You will need 10 discs for the light reactions and 10 discs for the dark reactions. Take one of the syringes and remove the plunger from it. Using the tweezers, carefully place 10 discs in the syringe. Be careful while using the tweezers as you do not want to damage the discs. Make sure that all the discs fall to the bottom of the syringe. You may use a stirring rod if needed to push down the discs to the bottom. You must do this twice, 10 discs per syringe, one for the light, one for the dark. Add 5 drops of the soap solution to the leaf discs in both syringes. This helps with taking out all the oxygen in the leaf discs and ensuring that all the discs will sink. Once the soap is added, put the plunger back into the syringe and make sure you do not crush the leaves when putting pressure on the plunger. Try to remove as much oxygen as possible. Place the syringe in the bicarbonate solution and fill the syringe to approximately 40 milliliters. Flip the syringe over so that the oxygen is near the top. Apply pressure and release the oxygen, but make sure you do not release the bicarbonate solution. Do this for both the light and dark syringes. Cover the top of the syringe with your finger and paper towels. Pull on the plunger towards you. This pressure will take the oxygen out of the leaves, which is what we see in the form of bubbles. Continue doing so until all the discs in both syringes sink to the bottom. Since all the leaf discs have both sunk to the bottom, we can now take the plunger out of both syringes and pour the contents of each syringe into separate 50 milliliter beakers. One should be labeled with light and the other one should be labeled with dark so we can differentiate the different syringes. Pour any extra bicarbonate solution into the light and dark beakers so that they both have approximately the same amount of solution in each. Cover the dark beaker with aluminum foil and place both under a fluorescent lamp. After approximately 15 minutes, you will start seeing results in the beaker that is not covered with aluminum foil. This is because it has started to photosynthesize again, resulting in the leaf discs floating because oxygen in the leaf discs has changed the buoyancy of each leaf disc, causing the leaf disc to float again. The reason why the discs floated was because the oxygen builds up and it changes the buoyancy of the leaf in the water. The leaf discs in the light floated because the light-dependent reactions were still able to continue as the photons of lights were still able to excite the electrons in photosystem 2, which means all the light-dependent reactions will continue as per usual, And the Z protein, which is part of the light-dependent reactions, will continue to split the water molecules to produce H plus ions for the electrochemical gradient and oxygen as a byproduct. The beaker that was covered with aluminum foil, however, was unable to receive the light photons, so only the light-independent reactions would continue to happen. Light-independent reactions only include the Calvin cycle, and the Calvin cycle does not produce the oxygen needed to change the buoyancy of the leaf. Therefore, the light-independent reactions did not cause the leaf to float. Some experimental errors that could have happened while doing this procedure could include when putting the leaf discs into the syringe, not all of the leaf discs fell directly to the bottom. Some of them stuck to the sides of the syringe, and when pushing them down, that could cause damage to the actual leaf discs, altering our data. Another experimental error would include the actual vacuum created with the syringe and the pump. Considering that there were two different syringes, it wouldn't have been possible to apply the exact same pressure to each syringe. Therefore, one syringe could have had more oxygen pulled out and more bicarbonate pulled into the leaf disc than the other one, giving that one an advantage or a disadvantage depending on if it was the light dependent or the light independent syringe. Some of the challenges of making a self-designed lab would include the actual measurements. A lot of the measurements we had to alter so that they would fit the circumstances of our lab and a lab that could be done at our school.